Physics 1, memory, spin, and anesthesia, which are partially related at least. We're going to have two talks, both from physicists, and it's a great pleasure to introduce the first speaker, Matthew Fisher from UC Santa Barbara, who will be talking about, are we quantum computers or merely clever robots? Let's give a round of applause for Matthew.
Okay, well, I sometimes feel like I'm neither a clever robot or a quantum computer, particularly when I get up on stage and try to do something mechanical. So maybe I'll tell you a little bit about myself. I'm a theoretical quantum physicist, been thinking hard about quantum mechanics for 40 years. I first took my quantum mechanics class, and I want to tell you a story that I've been working towards for the last 4.5 years, which is synthesized in this intentionally rather provocative title, Are We Quantum Computers or Merely Clever Robots? So the story, though, begins not with quantum mechanics. It begins with lithium. Lithium is an atom. It's the third simplest atom. And lithium is also a drug for tempering mania and treating bipolar disorder, discovered in 1949, approved in the FDA in the 1970s. And I've been very interested in psychiatric pharmaceuticals, partly because I've been taking them for 25 years. I have many family members, as I imagine some of you might as well, who takes psychiatric medications, and some of them are really amazingly effective. And I've always wanted to try to understand how on earth, in particular in the case of lithium, one atom can modulate the tenor of one's conscious state. So I set out to try to understand how does lithium work, and once I started to set out to do that, I realized that one doesn't know how any psychiatric pharmaceuticals work. But I had an idea. When I started learning about lithium, I found out that there is a rare lithium isotope, lithium-6, which is only at about 8% of naturally occurring lithium. And lithium-6 differs from lithium-7 in the number of neutrons in the nucleus of the lithium atom. So lithium-6 has 3 protons and 3 neutrons. Lithium-7 has 3 protons and 4 neutrons. For those of us who take lithium from the pharmacy, we essentially taking lithium-7, 92% lithium-7. But it's possible to buy from isotope companies purified lithium-6, 95% lithium-6, or even more purified lithium-7, 99% lithium-7. And I had an idea. Why don't we take some isotope-modified lithium, feed it to some rats, and look for any behavioral differences that that might occur. And this seemed like kind of on the face of it a rather crazy suggestion, because biochemistry is all about electrons, chemical bonds forming and breaking, and the nuclear spins are generally believed in biochemistry to play a very small, if not entirely negligible, role. Then in the most amazing day of my scientific career, I, quote, discovered an experiment that was done in 1986 at the Cornell University Medical School, where they basically did this experiment. They looked at the mothering behavior of rats who had been fed CVS pharmacy, if you will, naturally occurring lithium, lithium-6 and lithium-7 purified, and control rats. And I want to show you one table from this experiment, not to convince you that it's right, but just to whet your appetite, as it did for me when I first saw this 4.5 years ago. But to set this up, maybe let me ask, who here knows someone that has taken lithium? So that's the lot. Now here's a trickier question, and I'd be delighted if anyone can answer it. Who here has at one point taken lithium? Yay, yay, you guys. Yeah. If you're like me, you can be proud that you take an atom. The taboo about psychiatric pharmaceuticals has to start ending. It's a disaster. But if one takes lithium at high doses, it tends to damp down one's affect a little bit. It flattens one out. I have a family relative who visits from the East Coast who has serious bipolar disorder. When she's on low doses of lithium, her brain is rushing. When she's on moderate to high doses of lithium, she's flattened out. Indeed, when she gets off the plane, I can tell almost immediately what her lithium dosage is by looking in her eyes. So what about this experiment on the mothering behavior of rats? They looked at various behaviors: nest building, nursing, grooming of pups, retrieval of pups, grooming of self, state of alertness, and so forth. The second column there is the control, the rats that were fed no lithium. All their behaviors were average. That's a little worrying. Was this experiment done blind? Was it done double blind? And what does double blind mean when you're doing an experiment on animals? And if someone can explain to me later, please do. The third column is pharmacy lithium, lithium naturally occurring, essentially lithium-7, and the fourth column is 99% lithium-7. And in both of those columns, the words, and this is very subjective, of course, are the same. Nest building was absent. Nursing was infrequent, short duration. Grooming of pups was infrequent. Grooming of self was absent. State of alertness was low. Okay? Now, that's consistent with my experience of taking high doses of lithium, and I think other people's experiences as well. It just flattens your affect out. What about lithium-6? So this is the rare lithium isotope when purified. When I saw this column, the fifth column, it just blew my mind. So lithium-6 mothers, their nest building was excessive, their nursing was very frequent, long duration, grooming of pups excessive, retrieval of pups excessive. I'm not sure how you can be excessive with that, but maybe you're like a helicopter mother, perhaps. Grooming of self, average. State of alertness, as opposed to average or low, very high. That's the drug I want. And indeed, you can order it from an isotope company, and I could take the lithium I take now and switch it out. I don't dare do it. I've been trying to reproduce this experiment, do some experiments like this, and we've got 2 already completed. We need to do some more. But if I take at face value this result, which I'm not trying to convince you of necessarily it's right, but it's remarkable because one less neutron in lithium-6 changes the sign of the effect on the conscious tenor of these mothering rats. And if it's really true that it's the neutron which is important, a neutron is a quantum mechanical particle. It's as quantum mechanical particle as you can get. And so that, raised the question, could it conceivably be that we have quantum processing in the brain? Are we, in fact, quantum computers? Now, I'm certainly not the first person to ask this question, and it's been asked by at least 2 participants in this conference, and point out in particular Roger Penrose's book in 1989, The Emperor's New Mind. I just finished my PhD when that book came out, and I read it, and I would say I was really inspired. It was, a remarkable book, which I recommend to everybody. I reached the end, and I wasn't completely convinced, but I don't think, that was really even the intent. I did have some issues with the biological hardware, if you will, where the putative quantum effects were perhaps taking place in microtubules, and that I have issues with that. Now, of course, these both quantum healing and Roger's book met with a tremendous skepticism. The skepticism was myriad, and the skepticism was not unjustified. And the reason is biological processes are slow. So, for example, a protein folding rate is maybe 10 to the 4th hertz. Neuron spike firing frequency maybe 100 hertz. So one can take, in quantum mechanics, a frequency and convert it to a crossover temperature T star. It's a quantum crossover temperature, taking the frequency, multiplying by Planck's constant and divided by Boltzmann's constant to get this T star. And T star separates the regime where if you're cold, compared to T star, quantum effects are manifest. If you're warm, compared to T star, the quantum effects wash out. And if we take as an example a frequency of 10 to the 4th hertz, this crossover temperature is a tenth of a microkelvin, 10 to the minus 7 Kelvin. Of course, that's way, 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 way smaller than our body temperature of 310 degrees Kelvin. So the conclusion is biological processes are thermal, not quantum. The body is simply too hot for quantum effects. And I think 99% of the physicists would conclude this. But there is a loophole, and it's really a quantum loophole. The argument I gave assumes thermal equilibrium. But if we think about the worldwide efforts to build quantum computers in the lab, the whole point is to isolate the qubits. You don't want the quantum computer thermalizing with the environment. Quite the contrary. You really require isolation. And if a quantum computer does entangle with the environment too much, it actually degrades the quantum processing and sort of ruins the computation. So isolation is the key. Indeed, isolation of qubits is paramount in the various different hardware, superconducting qubits, solid-state qubits in diamond, and quantum dots, topological qubits, and so forth. There's many, many different cold atoms, ion traps, and so forth. And since isolation of qubits is paramount, it's also hard, and something that's hard is very expensive. And when things are very expensive in this present funding climate in the United States, the federal government doesn't fund it anymore, and our rich friends at Google and IBM and Microsoft, they have stepped up to the plate. And literally, both Microsoft and Google, they're putting in about $50 million a year is my guess, I don't know for sure, towards building or trying to build a quantum computer, either with superconducting junctions or topological qubits. And this is great. I think it's very, very, very challenging, and progress will be made. It will be slow, for sure. But I think there's more platforms than that are covered by these companies, and so I'm really trying to look outward. I'm not a social media type person, but I gather that Facebook is something that a lot of people use. They make a lot of money. So where's Mark Zuckerberg's platform? Mark, where is it? Why don't you pick one of these quantum computing platforms? Now, Twitter, frankly, before President Trump became president, I didn't even know what Twitter was. And now I wish I didn't. OK, we need isolation. Let's come back to biology and the brain. What is isolated from the warm, wet biological environment? There's only one thing which a nucleus spins. OK, protons and neutrons have nucleus spin a half, and some atomic nuclei, which are made of protons and neutrons, have a nucleus spin. And the nucleus spins, being protected inside the atoms, are very isolated. And to give you an idea, if you take sodium chloride, salt, and you dissolve it in water, the sodium ions come off. And you can ask, how long will it take for the nucleus spin of the sodium to decohere off the water? It's due to fluctuations of electric fields and magnetic fields. And the time scale is about 1/10 of a second. OK, so that's very long on sort of biochemical time scales, but it's not so long on human time scales. But these time scales vary. Lithium-7, which is a CVS pharmacy lithium, the decoherence time, which is longer, it's 10 seconds, 2 orders of magnitude longer, so the lithium-7 nuclei is more, the nucleus spin is more isolated. But then when I saw this result, it just blew me away. Lithium-6, and that was the nucleus spin, that was the atom which was purported to lift up the mothering behavior, the decoherence time is 5 minutes. Now, I don't know about you, but as I get older, I would say my memory is now slipping below 5 minutes. So that was, I said, that's incredible. There's the lithium-6 rat reading the dictionary. Still reads books, doesn't read Wikipedia yet. OK, so nucleus spin. So, this really suggested that conceivably quantum processing with nucleus spins might be operational in the brain. And so what I've been doing and exploring for the last 4 years is what I might call a reverse engineering approach. So imagine God comes down and tells you, I've cleverly designed chemistry and biology and neuroscience and evolution so that over 2 billion years, evolution has found a way to quantum process with nucleus spins cognitively. Reverse engineer it. Okay? Now, if you say, well, that's crazy, it can't be, then you're not going to be interested in trying to reverse engineer something that doesn't exist. But if you are willing to suspend belief, as I have done, which is saying maybe it does exist, and in fact, I'm going to assume that quantum processing is present cognitively, and then ask, can I reverse engineer? And if I can, maybe it actually is. If I can't, probably it isn't. So the way I did this is to start writing down a list of required conditions that would have to be satisfied by biology and biochemistry and neuroscience in order for evolution to have found this path to quantum process with nuclear spins. And here's just a few of those things that I listed. First and foremost, one needs a biological atom with a very isolated nuclear spin to serve as a neural qubit, a mechanism for quantum entangling pairs of those nuclear spins, a mechanism for quantum processing, like in a quantum computer, we need a quantum process, and even most important, a method for what I call quantum to biochemical transduction. It's not very helpful if we have a quantum computer in our brain, but it doesn't actually couple to the neurons that fire, and so we can think, oh, I want to raise my hand, and then actually raise it. Right? So then I had to sit down and say, can such conditions be fulfilled in biology and in chemistry? And so I had to do something which was very difficult for a physicist, which I had to sit down and actually learn something. Physicists, we know an enormous amount about next to nothing. Biologists and chemists know a lot about many things. So together with Wikipedia and Google, my collaborators, wonderful collaborators for the last 4 years, I've been trying to learn the biology and the chemistry to try to find out whether reverse engineering is possible. And what I want to do here in the remaining time is to tell you a little bit about that effort and where it stands presently. So the most important thing is a neural qubit. Now you might say, well, why don't we use lithium-6 as a neural qubit? Well, lithium is a trace element, so if you don't take it from CVS Pharmacy, the lithium concentration in your blood is very low. So I put lithium aside and come back to it later. So I said, okay, we need a common biological atom with a very isolated nuclear spin, which has a very long nuclear spin decoherence time. And learning about the NMR literature, one finds out that what that really means is we need an atom with a nuclear spin of 1/2. Spin of 1/2 is insensitive to electric fields, only to magnetic fields. And we need that atom with nuclear spin 1/2 to be in a small molecule that can tumble in water. For the physicists, that would be getting motional narrowing. And so I had to learn what are the common biological atoms. Well, the biochemical elements, chnops, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, all have one letter. That's nice. The bioelectrical ions, sodium, potassium, chloride, magnesium, and calcium I should really list in both, because calcium is sometimes said to be an electrical to chemical converter. So here are the common biological elements. Which, if any of these, has nuclear spin 1/2? Well, the proton does. Let's leave that aside. Do any of the others? Well, I didn't know, so I had to look it up on Wikipedia. And quite remarkably, there's only one of these common biological elements which has nuclear spin 1/2 besides the proton, which is phosphorus. Phosphorus there's only one stable isotope of phosphorus, phosphorus 31, and the nucleus of phosphorus 31 has nuclear spin 1/2. So this first step in attempting to reverse engineer a putative process of quantum processing in the brain, which may or may not be taking place, I've arrived at the fact that there's only one possible neural qubit. And if the phosphorus nuclear spin is not being used for processing quantum mechanically in the brain, I don't think the brain is quantum processing on human timescales. End of story. So this is it for me. But we need phosphorus in a small molecule that can tumble. Well, I had to start learning some of the biology that I never learned 40 years ago. But I did remember adenine triphosphate, adenine diphosphate, phospholipids. Like phosphates are all over the place. So what a phosphate ion is a phosphorus atom surrounded by a tetrahedral arrangement of 4 oxygen ions, schematically drawn there, and in the chemical way of drawing it as well. And so the first thing I asked, okay, well, what happens if you take sodium phosphate, dissolve it in water, so you have the phosphate ions floating around in water? How long does the nuclear spin, the spin 1/2 nuclear spin of the phosphorus, take to decohere, to entangle with the water? And the time is around a second. And this was disappointingly short. I thought it might be more like 5 minutes like lithium. And the reason it's not longer is because protons at physiological pHs near 7 are floating around, and this phosphate ion is negatively charged, and so the protons can and do weakly bind to the negatively charged oxygens, as I've schematicized there. The proton nuclear spin and the phosphorus nuclear spin entangle with one another. That's okay, but what's not okay is that the proton then hops off. It hops on and off, and that decoheres the phosphorus nuclear spin in the time scale of around a second. But I wasn't about to give up, so I asked, is there another biological ion, or cation, a positively charged ion, which could conceivably outcompete the proton in binding to the phosphate, the negatively charged oxygen of the phosphates? And I spent time learning about magnesium 2+, sodium plus, potassium plus. But it turned out there's really only one possible biological cation which can serve the function of displacing the proton and binding to the negatively charged oxygens, and that's calcium. And there are many hints of this. Bone is a mineral, it's a crystal, and it's calcium phosphate. And the story behind what I've called here the Posner molecule is quite interesting. In 1975, there was a bone scientist who was looking at the crystal structure of bone, and he noticed that inside the unit cell were these Posner clusters, things which were called after him, clusters, which are calcium 9 phosphate 6. And there were 2 of them in the unit cell, and then the unit cell was repeated. He suggested that maybe these calcium phosphate clusters might have some role in nucleation, but at that point in 1975, it wasn't clear whether or not these Posner clusters could exist as free-floating molecules in solution. Fast forward 35 years, and there were 3 or 4 experiments which basically gave extremely strong evidence that, in fact, these Posner clusters should be called Posner molecules because they can float stably in what's called simulated body fluids. So water with the salt concentration appropriate for your extracellular fluid. So, and very rough estimates of the nuclear spin, the phosphorus nuclear spin decoherence time in this Posner molecule, which is almost spherical, so it rotates very rapidly, gives times of order 10 to the 6th seconds. Could it be 10 to the 4th seconds? Yes. Could it be 10 to the 7th seconds? Yes. Very rough estimates. But in any case, it appears that this Posner molecule is really the ideal setting for quantum storage and processing. And I really should emphasize that I was looking for a calcium phosphate complex, and it took me about a month to find it because I was searching under calcium phosphate molecules, calcium phosphate complexes, and I should have searched under calcium phosphate clusters. In any case, there it is, the Posner molecule. Well, that's fine, even if it's there. In order to do quantum processing, one needs to quantum entangle different nuclear spins. And there are some biological chemical reactions. Adenine triphosphate goes to adenine monophosphate and releases what's sometimes called pyrophosphate. It's shown there. It's basically 2 phosphates joined together. And then there's another chemical reaction which takes this diphosphate and breaks it into 2 phosphates. And what's remarkable about that is that in the molecule, the diphosphate, the 2 phosphorus nuclear spins are entangled with one another. It's a bit like the hydrogen molecule where there's ortho and para states of hydrogen. And so once this diphosphate with the phosphorus nuclear spins entangled, quantum entangled, goes through an enzyme, shown here, and they get broken and they separate, the nuclear spins in these 2 phosphorus nuclei, even though these phosphates are separated, are quantum entangled. So released pairs of phosphates have 31p nuclear spins quantum entangled. So one can imagine then one has a quantum entangled shower, as depicted schematically here. You just have, creating a whole set of these. And maybe this is going on throughout your brain. Well, I made this joke before, so I apologize to those who have heard it, but it is going on throughout my brain because I cannot stop thinking about it. Create intermolecular nuclear spin entanglement. Quantum processing would occur with networks of quantum entangled Posner molecules. And quantum entanglement, it turns out, will or should influence non-local pair binding. So let me make the following comment. To these Posner molecules, the reason I believe that biology hasn't discovered them in 100 years is because they don't react with anything. And if there's a molecule that doesn't react, you don't see it. It's not quite true that it doesn't react with anything. There are 2 reactions. 2 of these Posner molecules can chemically bond to one another. That's a chemical reaction. And these Posner molecules can dissociate into their calcium and phosphates. That's a chemical reaction. And one can show, because of this quantum entanglement between these Posner molecules, that if, as I've shown here, A and A prime, these 2 Posner molecules are quantum entangled, B and B prime are quantum entangled. If A and B chemically bond together, then A prime and B prime are going to be more likely to chemically bond together, about 3 times as likely. So what one would have is quantum entanglement non-locally in chemical reactions. So you could think of this as like a Bell's inequality, except rather than having 2 spin ahas, you actually have chemical reactions which are going off in concert in some sense, which can happen non-locally. But we also need some sort of quantum to biochemical transduction. So if 2 of these Posner molecules bind together, they rotate more slowly, and in slightly acidic conditions, they're going to dissolve, release their calciums and phosphates. And in neuroscience, it turns out that the calcium ion is really the most important ion. And when released through calcium channels, when an action potential comes down, hits the synapse coming down the axon, calcium channels are open, and calcium, just 3 calcium ions have to bind to vesicles which store the neurotransmitters like glutamate or GABA, and which are then released into the synaptic cleft and can stimulate the dendrite of the postsynaptic neuron. So the picture one has here is that these released calcium ions are modulating a neural activity, but really creating a, quote, quantum network that coexists with the classical neural network. So let me step back. This is classical neuroscience, namely all neuroscience, the classical brain. And what I've been exploring then is what one might call quantum neuroscience, so these nuclear spins. And absolutely critical is this link between them. And you might want to think about it, the classical neuroscience is like the experimentalist in building a quantum computer, and the quantum neuroscience is the quantum computer that's being built, and you have to be very gentle in touching that, and the quantum computer and controlling it. But that's what I've talked about, that the nuclear spins can modulate chemical reaction rates. On the one hand, chemical reactions can implement measurements of nuclear spin. So these, using biochemical reactions that, fairly well understood, one can then get this, potentially this exquisite linkage between the classical neuroscience and this putative quantum neuroscience. So at this stage, what I've been telling is a story. It's a story based on, essentially all knowledge that I could find out there on Wikipedia and Google that's pertinent to my question of, can I reverse engineer putative quantum processing with nuclear spins cognitively? And I've found pieces of a puzzle, and in my head I can imagine perhaps an almost complete puzzle. And what we need now are experiments to discern the precise shapes of these various pieces of the puzzle, and then see if they start fitting together or not. One of the goals, short-term, goals, I can say 2 to 3 years, whatever that means, could be 5, is to try to synthesize a quantum brain. The approach here is basically the following. So genetic engineers, what they do is they use biology and all the clever biological proteins, which have evolved over 2.5 billion years, to do what they want to do. They snip bits of DNA out and put them into other bits of DNA and so forth. So if quantum processing with nuclear spins is actually operational in the brain, and that biology for 2.5 billion years has perfected that, we should be able to, using the biological enzymes and molecules and appropriate nuclear spins, build a quantum brain in a test tube, right? And the simplest quantum brain in the test tube that I want to imagine is we can generate nuclear spin entangled phosphorus 31. I'm really just taking you through some of the story. You have them in the test tube, these Posner molecules which are entangled. We add what are called calcium fluorescent molecules. We pour into 2 test tubes. Okay, now we imagine taking these 2 test tubes on the opposite side of a lab. But if this picture is right, they're actually quantum entangled. In these 2 test tubes, these Posner molecules, in fact, their rotations are quantum entangled with one another. We drop a little acid, hydrochloric acid, or maybe LSD. LSD is an acid. You could stick it in there, and maybe you'd believe you were seeing something interesting. But I want to try it with hydrochloric acid to see if I actually am. And what one would be looking for is in this acidic environment, these Posner molecules would melt, just like bone. I don't know if anyone saw Breaking Bad. But if you want to get rid of a body, sticking in a vat of acid, because it melts bone, don't do it on the 2nd floor. All right, let's see. So drop a little bit of acid. that would dissolve these poison molecules that release their calcium ions. They would bind to the calcium fluorescence molecules, and you'd get fluorescence. You'd get little flicks of light coming off these two test tubes. And what one would be looking for are cross-correlations of the fluorescence. Are these little flicks of light, have correlations between them, even if these test tubes are on the other side of the room? If any cross-correlated fluorescence is observed, it would really be direct evidence of quantum entanglement transducing a non-local chemical response. It would be really revolutionary in many regards, and it would give one some confidence, at least me, that perhaps I'm on on a reasonable track and would want to continue going. And moreover, if this worked, you could get thousands and thousands of these little test tubes, dishes, stirring things around and reacting and having fluorescence going on, and design some sort of liquid state NMR quantum computer. That's what one would be looking at. So I gave this talk a little while ago, and someone came up to me afterwards and said, "Oh, Matthew, there's a really nice quote from Isaac Newton. I can calculate the motion of the heavenly bodies, but not the madness of men." Well, okay, Newton died about 300 years ago. I think it's maybe time we address calculating the madness of men. And in some sense, that's what has motivated me to start with lithium and then continue on this quantum path. Now, in neuroscience, as some of you know, there's a little jingle, which is that neurons that fire together, wire together. I imagine most of you have heard of that. Jingles are kind of corny, and that's a little corny. Maybe it has some meaning. Neurons that fire together, wire together, but okay, it tells you something. So, here I was into the word jingle jangle. It turns out it's the 2016 Nobel Prize winner in literature, Bob Dylan. And a good friend of mine last year, won the Physics Nobel Prize and got the honor of actually going to Stockholm, which was, a real party. Bob Dylan didn't show up. It's probably good that he didn't, because he would have, taken all the wind out of the sails of all the other Nobel laureates. But his medal was sitting right there, and we got to snap a picture of it. I wanted to actually just steal it. There it is. It was just on a table. But that's Bob Dylan. So where is the Jingle Jangle? Well, there's a song, Mr. Tambourine Man. I'm not a very good singer, but hey, Mr. Tambourine Man, play a song for me. In the jingle jangle morning, I'll come following you. And then jingle jangle, it's not in the regular dictionary, but it's in the Urban Online Dictionary. And it reads, jingle jangle is a description for something that is beautiful, easy, carefree, and or simply wonderful. And if our neurons are being modulated by quantum processing of nuclear spins in a quantum coherent way, that'd be kind of beautiful, maybe easy. I don't know, it's kind of easy to think sometimes. Certainly wonderful. So I think that is all I wanted to say. And I'm viewing this as just the beginning of a journey. And the journey has, at this stage, been essentially one of building a conceptual framework using known experiments in biology, biochemistry, chemistry, organic chemistry, neuroscience, psychiatry, piecing them together in a story. And now we're going to turn the corner and start actually doing, we've started to do some experiments. We really need to push hard on the experiment. So let me stop there, and thank you for your attention.
Thank you. Thank you very much. That was very interesting. We'll have time for some questions. Let me, I'll take the prerogative. Jack, we'll get the mic in a second. I have to make a comment, though. Lithium does stabilize microtubules. That is well known.
I was going to mention microtubules.
Yeah, go ahead.
In my question and comment. So where calcium and phosphorus come together actually is in microtubules.
Use the mic.
Yeah, we wrote several papers on this topic of memory, molecular memory formation using microtubules by phosphorylations. So phosphorus phosphorylation using calcium calmodulin kinase enzyme. So you have calcium as a trigger, and then the effect is phosphorylation of especially serine amino acids on the C-termini of microtubules. And these are very dynamical structures, so actually you can have interactions.
Let me interrupt. I don't buy it. I don't buy it. Maybe phosphate and phosphorylation is going on everywhere.
Exactly.
In the mitochondria, there are in fact sludges of calcium phosphate, and the calcium is being regulated and so forth. But in order to have quantum processing operational on human timescales, including quantum memories, basically, over years and longer timescales, I don't see how any degrees of freedom that are bigger than a nuclear spin can be coherent for longer than, I don't know, 10 to the minus 13 seconds. And so I think that's a fundamental problem with microtubules or any other putative hardware.
You're not allowing me to.
No, I'm not, because I'm not. Yeah. Okay, well,
There's actually evidence for 10 to the minus 4 seconds in microtubules.
Okay, well, is that enough? I don't know, my memory is longer
yes,
than that.
it is, actually.
Okay, good.
Okay, thanks.
Somebody else?
Tough crowd.
What?
Dr. Fisher, fascinating detective story. Thank you. You just told it so well. My question is that if the bottommost layer is, as you say, classical neuroscience, and there's can you understand me?
If there's what? Sorry, I missed it.
The bottommost layer is the classical neuroscience,
Yeah.
right? And there's a transduction from the quantum that you just discovered. Is it possible that there's another layer between information in the universe and tapping that to our biology? And are you familiar with some of the work that Anirban Bandyopadhyay is doing on this hierarchy of frequencies?
Well, okay, let me make one comment. I mean, within this story, the most important aspect of the phosphorus nuclear spins is that they have spin half, but that they're fermions. And fermions are the most strange particles, and there's a lot of non-locality in fermions. I mean, the fermion creation operators don't commute, even when they're far apart. And so, if this process is present, it's using basically the fermions, which are these fundamental particles in space-time, to quantum process with. And I think maybe that's all I can say on that.
Okay.
Hi, Matthew. Thanks for the talk. It was great. I saw your next slide, and it was the one thing I wanted to ask. So, what happened to the lithium? How does this relate back to lithium?
Thank you. Okay, so there's a graduate student who's been helping me a little bit in calculating the energy of these Posner molecules. There's 9 calciums and 6 phosphates. The 9 calciums 8 of them sit on the corners of a cube. The 9th is in the center, it's like body-centered cubic, and then there are 6 phosphates on the 6 faces of the cube. And so, I asked the graduate student, well, why not consider the 2 situations, one with a Posner molecule in water and with 2 lithium ions floating in water, and you have to look up the enthalpy of hydration for lithium ions. Compare that to taking the central calcium ion out of the Posner molecule and sticking in 2 lithium ions, and then looking at the hydration of enthalpy of calcium, and ask, is that lower in energy or not? And from his calculations, it lowers the energy by around an electron volt, which is, 40 or 50 kT. So if that calculation is right, it would actually say that if there's enough lithium concentration available, in particular, concentration comparable to the calcium concentration, which is indeed, when you take lithium therapeutically, it's right around 1 millimolar, and calcium is a couple millimolar. So the idea is that if these lithium ions get inside these Posner molecules, particularly lithium-7, which would be decohered when the molecule rotates due to electric fields, that would really mess up. It'd be deleterious to storing quantum information and doing quantum processing. Now, do lithium ions actually get inside these Posner molecules? Well, we can do experiments to try to look and see whether they do or not. And if they do, we will be able to see that they do. So. Yeah, well, lithium-6 is great. I mean, so lithium-6, it's not sensitive or relatively insensitive to electric fields. And when this molecule rotates, the electric fields are fluctuating very, very rapidly. So lithium-6 will be largely immune to that. On the one hand, but on the other hand, lithium-6, its nuclear spin will couple to the phosphorus nuclear spins, and one will have a larger Hilbert space for the quantum system. It's like, a bigger system. Now, could that be helpful in some way? Could lithium-6 actually be being like co-opted? I don't know, possibly. But that's the story at this stage. That I put lithium aside for 4 years. I didn't know. I should emphasize, when I started this, I wasn't looking for quantum mechanics in the brain. I was looking at trying to understand lithium. So, anyway.
Last question.
So how long do you think it will take for two spins in two different nuclei to entangle? So what's your gate time?
How long does it take to entangle or to decohere?
No, to see what one always has to look at, what is my gate time in relation to the
yeah,
coherence
I
time.
understand.
So
Yeah. Well, okay, so let's say the decoherence time is 10 to the 6 seconds.
which
Okay? Which
very
it may
long.
well be. And then the gate time, is, nanosecond. So it probably has like 15 orders of magnitude, which is a little bit better than the ion traps and the cold atoms and so forth, where people are struggling to get a factor of 1,000.
Yeah,
So is 15
exactly.
orders of
You
magnitude long
wouldn't
enough? It
need
might
to-
be.
Yes, that's what I'm getting at. You don't necessarily need very many gate operations and not too many qubits to do something already rather interesting. So a second would be plenty to do, or
Well,
a fraction of a second.
I don't know. I don't think so. I mean, I think, look, I would like memories to be actually stored quantum mechanically. Those memories that we access consciously, and those memories, last for years.
Okay, I want to point out it's a unitary mechanism of anesthesia with a question. I mean, it's still open. We're looking at this. I'm a physicist by training, but I work at Nova Southeastern University, situated in 3 unique departments: neuroscience, computer science, and immunology. And primarily, I look at neuroinflammatory illnesses from a physics standpoint, building models. So this is work I've been working on for close to 10 years now with my former PhD supervisor, Jack Tazinski, and Stuart Hameroff. So when I was finishing up my undergrad and moving on to grad studies, I fell into the world of consciousness and the relation to physics. And at the time, I didn't know much about it, but what I quickly learned is that there's one thing that we know that affects consciousness directly, and that's anesthesia. So let's get into that. What is general anesthesia? Anesthesia is defined as any drug that provides these 4 things. Analgesia, it blocks the conscious sensation of pain. Hypnosis, it produces unconsciousness. It prevents memory formation, and it prevents movement, because you don't want someone moving around on the table when you're cutting them open. Now, depending on which textbooks you're reading and when they were printed, you may see some of these removed. I think more recently they've removed the term hypnosis, producing unconsciousness. I don't know what the reason is for that. You may want to talk to Stuart, but I'm not quite sure. They're medical marvels. They're required for all modern surgery. I mean, you wouldn't want to go under a knife without some sort of anesthesia. But we still don't know how they work. We don't know what they do, besides knock you out. So why do we need to know this? We have a bunch of anesthetics. We can use them. We use them in surgery all day long. But why do we need to know how they work? Most anesthetics have been determined through serendipity. I mean, accidentally discovered. You're trying them out for something else. You try it out in an animal, and the animal gets knocked out, and you're like, oh, that's great. Let's test it out, and eventually it becomes an anesthetic for humans. One, from a philosophical and scientific standpoint, it's the only known probe of consciousness. Okay, we can turn it on, turn it off all day long, and this is eventually what got Stuart interested in consciousness studies. But from a more practical perspective, we want to know how they work for reasons such as postoperative cognitive dysfunction. So what's been known for a while is that if you have overexposure to anesthesia, what happens is when you wake up, you have a slight cognitive dysfunction. And depending on your age and your condition, it takes sometimes days, could be weeks, for this cognitive function to come back up. Now, for most people, that's not a problem, but for people who already have a neurodegenerative or a cognitive condition, this can be quite detrimental. It can exacerbate the problem. So if we figure out how anesthetics work, potentially we can design new anesthetics that may be free from these harmful side effects. Well, what do we know? What do we know about anesthetics? One, if you look across anesthetics, we know that they don't share a structural similarity. I mean, we can look over here. We got things like nitrous oxide and dialkyl ether, cyclopropane, completely different molecules. There's related families. We have the halothane, desflurane, the fluorinated ethers, or fluorinated hydrocarbons. But these things range across a span of about 35 orders of magnitude in molecular volume, starting from things like a single atom, xenon, which is a very good anesthetic, to things like steroids, 56 atom steroids. So from 1 atom to 56 atoms. Right? So how does that work? I mean, they don't share any shape. They obviously don't look the same. However, conversely, even though they don't look the same, they're not shaped the same, they're not the same size, they do have a relation that holds strongly, and this is known as the Meyer Overton correlation. So around the turn of the century, the last century, around 1900, Meyer and Overton separately discovered that the potency of anesthetics correlates strongly with their solubility in a lipid environment, like an olive oil. And so this suggested a unitary mechanism, something to do with the way that they dissolve. This correlation holds across multiple species, starting from single-celled organisms like paramecia. You can stop their cytoplasmic streaming and so forth, all the way up to human beings. It's also been observed in plants, but I'm not going to get into plant consciousness or anything like that. So we're going to stick in the animal realm. Now, at first, because this is a mechanism that suggests something to do with solubility in oil, the first proposed unitary mechanism obviously related to lipids. So they suggested that anesthetics act by binding in the lipid membranes and causing them to become more fluid in order to alter neuron function. So at the time as well, they thought that neuron function had to do with the membrane itself. So altering that could change the way that neurons fire. But there's inconsistencies in this lipid model. One, okay, the lipid environment, the bilipid membrane, is a achiral environment. It doesn't matter which way you sit in there. Everything looks the same as you look around. Okay, that's assuming that you're talking about the same types of lipids in there. Okay, you can make an argument that you might have different types of phospholipids in an arrangement, but I'm not sure of evidence for that. But anesthetics themselves are chiral. So if you look at anesthetics, if you take this molecule here, this is isoflurane, and you have a left-handed version and a right-handed version. They're mirror images of one another. The exact same chemical properties, the exact same solubility in olive oil, okay? It's made up the same thing, but their anesthetic effect is completely different. So one is a potent anesthetic, the other is almost useless. Okay, so there's some sort of chirality involved. Additionally, there's things like what's known as the cutoff effect. So things like N-alkanes or N-alcohols, if you take one of these structures and you keep adding chains to it, you can make them longer, and the longer you make them, it changes their solubility in a particular fashion. And it predicts a particular trend then for their anesthetic potency. But what ends up happening is when you get to a certain length, after that, they lose all anesthetic effect. So there's no increase by making them longer. So this is known as the cutoff effect, a lack of anesthetic effect, even though it should be following the Meyer Overton correlation. Thirdly, there's other exceptions to the rule. These are small molecules, right along the same size as anesthetics, but when you bind them into, you look at them, they have the same solubility, but when you actually give them as an anesthetic, they don't do anything at any concentration. They don't have any anesthetic effect. So this led Franks and Liebs to predict and demonstrate that maybe the anesthetics are working in some sort of chiral environment, like membrane proteins. And they were looking at membrane proteins because at the time, everything was looking at the membrane, and you thought you could directly influence neuron function by affecting the ion channels and the receptors. And so over time, people have been looking more and more at ion channels, and there is evidence that anesthetics do bind to these. Primarily, you have GABA, nicotinic acetylcholine receptors, serotonin receptors, glycine receptors. There's a little bit of there's stuff on sodium receptors or sodium channels. However, there's ambiguities in these. So you get things like you would expect an anesthetic to excite an inhibitory receptor, but instead it inhibits the inhibitory receptor, or it excites an excitatory receptor. Or you get certain anesthetics will bind to 3 out of the 4 membrane proteins, but it doesn't even act on the 4th one. So some will bind to some will bind to the other one, so there's varied inconsistencies. So this led people in the field of anesthesia to abandon the idea of a unitary mechanism. They deemed it fruitless. However, and we would get to microtubules eventually, microtubules are also on pathway targets for anesthetics. So this was originally suggested about 25 years before Meyer Overton found the lipid correlation. So Claude Bernard first looked at anesthetics working in cells, and he saw that it affected their cytoplasmic streaming. In the 70s and 80s, it was shown that volatile anesthetics like halothane can affect microtubule polymerization at very high concentrations. It completely obliterates them or causes them to turn into aberrant formations like macrotubules. And even more recently, the Eckenhoff group out of the University of Pennsylvania has shown that tubulin expression is altered, so gene expression of tubulin is altered by things like desflurane and sevoflurane. They've also shown that anesthetics like halothane and anthracene bind specifically to tubulin, so a direct interaction. They haven't done this for everything yet. I mean, one day we could hopefully look at this. But there's also other indirect evidence, like taxane-treated patients. Taxane is a microtubule stabilizing agent. So if you're stabilizing a microtubule via taxane, so someone who's undergoing chemotherapy, they're more resistant to anesthetics. And this is something that just recently came out. And my most favorite experiment is they looked at detectable proteins in rat neurons, and they exposed it to two types of anesthesia, halothane and isoflurane. And of the 500 detectable proteins, both membrane proteins and proteins within the cell, they found that tubulin was among the 2% that was affected by halothane, the 1% that was affected by isoflurane, and one of 3 proteins that was affected by both. The other two, one is a heat shock protein, but I can't recall what the third one is. Now, I'm not sure if everyone knows what the microtubule cytoskeleton is. You may. I'll just give you a quick primer. The cytoskeleton, it forms these elaborate networks in your neurons. Microtubules are in all eukaryotic cells. They're everywhere. But it's unique in the neuron because it forms these unique architectures that are stabilized by certain proteins to form the architecture of your synapses, your dendrites, your axon, and it maintains this morphology. It traverses cargo all throughout the cell, so it's responsible for moving proteins around and ion channels and anchoring them within the membrane. It's involved in learning and memory, so when when you're undergoing learning, there's some sort of reorganization of the microtubule cytoskeleton. This is work by Nancy Wolfe. As well, cognitive diseases have been shown, a lot of them, Alzheimer's, Parkinson's, dementias, bipolar disorders, schizophrenia, all show a dysregulation of the neuronal cytoskeleton. Okay, so is it the cause? Is it a symptom? That's still up for debate. Microtubules themselves are polymers of proteins. So there's a protein called tubulin. It's made up of 2 monomers, alpha monomer, which is gray, the beta monomer, which is darker gray. These things stick together end to end. They form like long tubes called protofilaments. These protofilaments stick end to end and they wrap around to form a nice hollow tube. In regular cells, these things turn over and grow and shrink. But in neurons, these things are stabilized by microtubule-associated proteins. Okay, so if anesthetics affect these microtubule proteins, these tubulin molecules, where do they bind? Okay, we know halothane, we know anthracene bind directly to tubulin, but where? The answer, we don't quite know. I mean, we have predictions. So several years ago, back in 2012, we had a paper where we simulated where anesthetics would bind. So what we did was we took the crystal structure of tubulin, we did a molecular dynamics simulation to capture 5 nanoseconds of this thing shaking in normal environment. And then we took that, identified dominant shapes of tubulin, and then looked for hydrophobic pockets that held the environment that anesthetics would like to bind in. So we looked inside these pockets, and we looked to see where the most probable place for anesthetic binding would be. We found 47 distinct sites. However, of those 47 sites, only 9 of them existed for about more than 70% of the 5 nanosecond simulation. The rest were quite transient, less than 50% at least, or at most, I should say. Of the 9 key sites, one is the GTP binding site. This is responsible for dimer stability, so alpha monomer stuck to beta monomer for keeping that strong. The colchicine binding site, colchicine is a known microtubule depolymerization agent. and it competes with halothane. It's known to compete with that, so that's interesting. The Vinca alkaloid binding site, but it was not found in the Taxol binding site, which is a microtubule stabilizing agent. We performed microtubule polymerization experiments, and these confirmed that halothane did not compete with Taxol to alter the Taxol stabilization of microtubules. However, it produced a slightly different polymerization scheme without Taxol present, and this is consistent, the lower line being normal control, the green line being halothane present. That is consistent of aberrant formations of microtubules. So that's cool. Okay, they bind there, you may know where they bind, but what do? Okay, it's believed that anesthetic effect is related to hydrophobicity or lipid solubility, but it's also related somehow to their permanent dipole strength, if they have one, or their polarizability. Now these hydrophobic pockets that are predicted to be where anesthetics bind, they're filled with non-polar amino acids, okay? These non-polar amino acids, they don't want to be in a lipid environment. They want to hide away from them, okay? And these aromatic amino acids primarily are very polarizable, okay? And they interact with one another via van der Waals forces. Now van der Waals forces are very, 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 very weak, okay? They're one of the weakest forces that we know, well, except barring the really small nuclear forces and whatnot. But in biology, they're still very important. Okay, so protein folding and the way that a protein is held together, even though they're small, there's many, many, many of these interactions, so they add up. In 1998, Stuart Hamerhoff proposed that anesthetics may work to inhibit these forces, and this became known as the quantum mobility theory of anesthesia. What is van der Waals dispersion or London dispersion forces? Normally, if you look at an atom, primarily it is over time, averaged over time. It is the electron distributions are symmetric. Okay? It looks like they're evenly distributed. But instantaneously, if you look at them, there may be some sort of asymmetry. And this gives rise to what's known as an instantaneous dipole. Okay? Now, there's nothing around that doesn't mean it doesn't do anything. But if there's another molecule nearby, what it can do is it can induce a dipole in there. And now you have this dipole-dipole interaction. And because dipoles that are oriented this way stick together, this forms an attractive force, and it bonds them together. And then they bond together and they oscillate. And then they can pull in a third thing and so forth. And you can do different configurations of the way that these dipoles are interacting. Can anesthetics affect this? We decided to look at this. We used benzene as a surrogate for aromatic amino acids. So benzene is a ring structure. It's a nonpolar structure. It's when you put them together, they attract via London dispersion dipole forces. So we calculated what this was and looked at the difference between whether the dipoles are pointing to the left or pointed to the right. And then we came and we stuck an anesthetic in there with its polarizability. And then we calculated out the effect of the anesthetic on the dispersion forces. And we found that the presence of halothane can shift this dipole oscillation frequency. Okay? In this case, we found that it increased the frequency, so you need to have more energy for the system to oscillate. However, we're only looking at 2 benzene rings, in this case in an ideal situation. So the quantum mobility theory of anesthesia kind of says that you have this microtubule. Within the patches of the microtubule, you have tubulin, which is filled with aromatic amino acids. The anesthetics bind within these regions. Normally, these dipoles oscillate back and forth, but when you stick in an anesthetic, it may damp these down. But is this a unitary mechanism? Okay, is it something that can explain anesthesia, or is this just something that happens with any drug? So if this is a unitary mechanism, it should distinguish between anesthetics, shown here in blue, non-anesthetics, which follow Meyer Overton but have absolutely no anesthetic effect, which is shown here in this red, and what's known as an anesthetic convulsant, which is shown here in green. So anesthetic convulsants, at a certain concentration, at one level that's low, when you give it, the patient will undergo convulsions. So they shake around. So it makes it a very bad anesthetic. But if you continually ramp up that concentration, eventually it will lead to anesthesia. You still don't use it. It has anesthetic properties, but there's something there that causes convulsions before you get to anesthetic effect. So they have anesthetic effect, but there's a problem with them ahead of time. Polarizability, so that ability of the dipoles to shift or to interact in van der Waals interactions, also seems to correlate well with the minimum alveolar concentration. So here we have, what is there on the left? The oil-gas partition coefficient. So this is inverted. So here we have solubility in olive oil along the y-axis. Along the x, you have the concentration in order to reach anesthesia. If you look at polarizability, you also get this nice curve here. However, what's interesting here is that you have these non-anesthetics that fall off of this line. And it seems to be some sort of discriminating factor. Oh, polarizability can predict non-anesthetics. But if you think about this, these concentrations for the non-anesthetics are derived from the Meyer Overton correlation. Because they don't cause anesthesia, there's no minimum alveolar concentration for them. We only predict it based on the Meyer Overton curve. So all this does is it shifts where their minimum alveolar concentration is, which shifts it to a lower concentration in order to line up on the line. So we'd shift it down here. And we know that's not true. Okay, so even if you give it at one concentration, it doesn't do anything. If you give it at a smaller concentration, it's still not going to do anything. However, there seems to be something here with polarizability. So what we did was we took tubulin again, we docked all these different anesthetic molecules, the non-anesthetics, the anesthetic convulsant, to those 9 prime sites on tubulin using docking calculations. We then calculated out the different van der Waals oscillation frequencies within tubulin in the absence and the presence of these different molecules. So what we get here is, because if you want to think about it, if you have several molecules, you can have oscillations. If you have 2 dipoles, they can oscillate back and forth this way, or they can oscillate opposite one another, depending on how they arrange with one another. So you can get these different modes of oscillation. And that's what's normally in tubulin, that's what's shown in these gray lines. Now for each of the addition of an anesthetic, because it's an additional dipole, you can get a new normal mode by adding in the anesthetic. And so here on the left, this is where you see the non-anesthetics, and over here on the right, this is where you see the anesthetics and the anesthetic convulsant fluorophil. I'm not quite sure if that has any meaning yet, whether low frequency changes are related to non-anesthesia, high frequency changes are related to anesthetic effect, but we're looking into it. So it seems to discriminate between non-anesthetics. So here if we plot this, the anesthetics here and the anesthetic anesthetic convulsant fluorophil here on the line, we get a nice agreement. And what this seems to suggest is that these non-anesthetics fall off the line. But again, we can manipulate their MAC because it's predicted from Meyer Overton. But even here, as we shift this, we find that it does not line up on the line because it has this cutoff as you get down here to the bottom. So it doesn't matter what you set the MAC at, these non-anesthetics don't seem to fall on it. And remember, this is a log scale. So it looks like a small difference here, but it's actually quite large. Oh, sorry, that's not a log scale. It's a small difference. However, this may just be curve fitting. Okay, we may just be fitting a curve to this thing. And when I heard someone from a famous physicist, he said a while ago that curve fitting is the lowest of the low for physicists. So maybe that's what we're doing here. But in addition to these new normal modes that are being introduced, those natural normal modes, those gray frequencies, they are also shifted by the presence of a molecule, but not in the terahertz range, which we're seeing here, in a much lower frequency range, in the gigahertz range. And so we look at those. So here's each of the anesthetics. It might be a little hard to see. So this is where the frequency of the normal mode, the natural normal mode exists, and this is by how much it's shifted by the presence of the anesthetic. And what you can notice is that the prime peak, the dominant peak for each of the anesthetics falls at almost the exact same spot, around 610 terahertz. Even for fluorophil, the anesthetic convulsant, it also falls around this 610 terahertz. However, for the non-anesthetic, F6 and trifluoromethylbenzene, at 610, you do not get a peak here. In fact, it doesn't shift it down, it shifts it up. So you get something different for non-anesthetics compared to anesthetics. So this downward shift now correlates with MAC, and it selects between non-anesthetics and anesthetics. Now this is something that we're still examining. It suggests a unitary mechanism of anesthesia. It supports the idea of quantum mobility theory. What this can serve to do, if you're shifting down these frequency modes, these frequency modes are most likely responsible for how proteins interact with one another. So if you have a protein oscillating at one frequency and another protein oscillating at the same frequency, then they might likely bind, and it may improve their connection. But if they're off resonance, you may get dissonant effects, and it could weaken the interaction. Okay, so this effective anesthetics may serve to weaken the microtubule polymerization. However, we're talking about at concentrations where we do not destroy the microtubule. Eventually, if you get higher and higher concentrations, these anesthetics are known to cause the whole microtubule to fall apart, but at clinical concentration, it doesn't destroy them. Well, we've done this in tubulin for obvious reasons, because that's the system we tend to work in. It remains to be seen if this is unique to tubulin. As well, we want to look at other anesthetic molecules. We've only been looking at inhalational molecules. We also want to start looking at the intravenous molecules like propofol and ketamine and whatnot. And we predict that we would get a similar effect, okay? We also want to test this out in other targets for anesthetics. We want to look at the GABA receptor and the glycine receptors and acetylcholine receptors and see, do we get the same effect, or is it something different? And if it's something different, then it shows that this kind of effect is unique to tubulin, and it may point to an actual effect of anesthetics directly on tubulin, a unitary mechanism. So that's something that we're currently working on. As well, we want to look at psychoactives. Okay, so that's the other end of the spectrum. Anesthetics knock you out. Psychoactives open you up. So if microtubules are the site of consciousness, what you would suspect is that psychoactive drugs should affect tubulin in some way. Okay, it should bind to tubulin, it should affect something in there some way. So if it's this van der Waals oscillation frequencies, you'd affect that the polarizability of these psychoactives could affect it in some way. How should it affect it? Maybe it's the opposite effect. However, then you would say, shouldn't trifluoromethylbenzene and the non-anesthetic F6, should they not also be psychoactives? Maybe it's because they're not affecting it to a great enough extent. Maybe you need to ramp it up. Or psychoactives could all work in a different frequency band. So you get the same effect somewhere else, but in a different frequency range compared to anesthetics. This is something we're looking at. Now, as a quick aside, we heard Stuart talk about this. I just wanted to say that lithium does stabilize microtubules. I'm just saying that. But if there's some sort of nuclear spin involved in consciousness and brain processing, if this mechanism is to hold true, then lithium would have to somehow, the nuclear spin would have to be involved in here some way. So there'd have to be some sort of spin-spin coupling via van der Waals forces or something along those lines that could account for the difference in the nuclear spin of lithium. And if that's not there, but lithium alters mood, then maybe it doesn't act via microtubules. But I don't know. It's something we're continually looking at. So in summary, anesthetics do affect ion channels. They do get into lipid membranes, but microtubules are also on pathway targets of anesthetics. Binding of anesthetics to microtubules occurs in hydrophobic pockets containing highly polarizable aromatic amino acids like tryptophan, tyrosine, and phenylalanine. The binding of these anesthetics alters the collective oscillations of van der Waals London dispersion forces. It adds new normal modes into the regular oscillation frequency of tubulin, as well as it downshifts a specific normal mode in the gigahertz range, where non-anesthetics do not follow that trend. Finally, changes in these oscillation patterns may affect overall polymerization rates of microtubule dynamics, and this may contribute to things like postoperative cognitive dysfunction by further weakening an already compromised cytoskeleton, as well as maybe lending itself to the loss of consciousness via the mechanism of quantum mobility theory. So I want to thank my team up at the Institute for Neuroimmune Medicine at Nova Southeastern University. We're primarily here in the clinical systems biology group. My collaborators, Jack at the University of Alberta, Stuart at University of Arizona, and Philip Currian at Howard University. Some funding agencies. Thank you very much.
Thank you, Travis. We have time for a few questions. Eric, anybody over here?
Oh, hey, Travis. So you pointed out these effects of binding tubulin in neurons. with anesthetic gases. Are there any other post-anesthetic tissues that would be affected by anesthesia that these binding effects would make apparent, such I'm thinking like temperature changes or other things somewhere else in other vulnerable cellular structures where tubulin is important?
It could. I mean, I'm not sure how the concentration which is in your lungs compares to what gets into your brain versus other organs. Stuart would probably be better to answer that. But if you're getting anesthetic anywhere in your body, it's going to affect the cytoskeleton there. And so if you have something like compromised cytoskeleton anywhere else, or it may affect liver cells or whatnot, it will affect microtubule dynamics anywhere. Okay, so this might have implications for people undergoing cancer or chemotherapy, because I mean you have one cancer cells divide rapidly, and the thing that pulls apart the genes in cell division is the microtubule cytoskeleton. It's unique in these other cells, it's not in neurons, to pull apart the genes. Now, generally chemotherapeutic avenues, they tend to target this structure and either stabilize it so it can form, but then it can't unform, so it gets stuck in that phase, or it depolymerizes them so that they can't pull them apart generally. So what you would expect then is if you're undergoing chemotherapy and then also undergoing some sort of anesthesia, that your anesthetics may interfere with your chemotherapeutic regimen.
Let me just say, there's anesthetics are all over the body, especially fat stores. I often tell my residents that there's more anesthetics in the patient's rear end than his brain, but the anesthetic's acting in the brain. And maybe Tony can comment on this. There are anesthetic effects at high doses, at least, on immune function and so forth, but it's usually not. Yeah, let him comment, then we'll come back to you. He's an anesthesia expert also.
All right, actually I had a question, so related to that. So obviously inhalational anesthetics affect the entire brain. Right. And they change neural activity in a heterogeneous manner throughout the brain. And maybe that's not important. Maybe the microtubules are the only important, but still, they everywhere. So is only one region important? So does it matter what brain region is affected in terms of its microtubules?
Well, it may. Okay, so it's true. So the anesthetic is going to go anywhere, just like the question from Eric here. I mean, you can go anywhere in your body, you can go anywhere in your brain. But there may be specific neurons to which, this quantum mobility theory that it's set up in such a way to tune it properly. For example, maybe your pyramidal cells, they may be more susceptible to this kind of effect. Okay, so likewise, going the other way is maybe it affects all brain microtubules in the exact same way, but because it's hitting everything all at once, then you're getting rid of only some of these may be responsible for consciousness. And I think Stuart talks about that in a thing that it may be the way that they're structured, so that the way that they're spaced, the way that they're connected to one another, there may only be certain regions that allow for things related to consciousness, the prime neurons. And when it affects those, it causes loss of consciousness, but it also affects everything else the exact same. Yeah. Does that kind of make sense to you?
As you know, anesthesia is fairly selective because the brain is quite active under anesthesia. Evoked potentials, EEG is slowed, and we used evoked potentials in spinal cord surgery. The patient's unconscious, but the brain is still processing signals from the extremities. So it's fairly selective. Maybe it's only affecting quantum effects on microtubules and not all the other stuff microtubules are doing, but that's just speculation.
That's what I was trying to say.
I think it's implicit in what was just said that there may be changes in the firing patterns of an anesthetized neuron, and I was wondering if you can speculate on the mechanism by which that would be changed.
Right, this is a little beyond my area, but what I do know is that consciousness and other brain processes are related to synchrony within the brain. Right? And these things switch from synchronous behavior to asynchronous behavior. And I think it's groups out of Michigan that have shown when you give anesthetics that it can change the rates of synchrony or throw things off synchrony in a certain degree. Now, if microtubules are responsible for somehow tuning whether things are responsive or unresponsive, and you change their fluidity by adding an anesthetic, they may be less likely to fire at the proper timing, And what that would give rise to is changes in the overall synchronization of the neuron firing in the brain.
Let me just make a comment. Firing and synchrony are not the same. Firing refers to axonal firing. Synchrony comes from the dendrites and soma, which activity there gives rise to firing. Anesthetics don't particularly affect the ability of a neuron to firing. They seem to act on the dendritic and somatic side.
My comment really is something that ties together the two talks that we've seen, and it's about lithium. I'm a psychiatrist, and I prescribe lithium for people, and one of the things that I was curious about is whether or not anybody's looked at lithium-6 and lithium-7 and the stability of tubulin, because it'd be very interesting to see whether or not there's a difference there. In looking at those rats that were given the lithium-6, I mean, it was almost like they were made manic because their behavior was excessive. And one of the complaints that people make about lithium when it's prescribed is it decreases their creativity. So it kind of suggests that maybe, if you have too much lithium, too much stability with the tubulin, maybe that decreases our process of creativity, and it could be related. So it's
Well, I know that the way that lithium works on microtubules, it can stabilize it. It competes with another ion, magnesium. So magnesium, it's required for microtubules to form and form their shape. Now, if you take that out and you put it in a lithium ion, lithium is a monovalent ion, magnesium is a divalent ion, and so it alters the way that it forms. Now, if you have two lithium ions in there to counteract the one magnesium, again, you can re-establish the same sort of ionic contribution, but the overall function is going to be different because now you're dealing with lithium as opposed to magnesium. So it has something to do with the magnesium contribution in microtubule formation. Okay, but no one, as far as I know, has even looked at this. I mean, the first I've heard about it is from Matthew saying that lithium-6 works differently than lithium-7. No one has, as far as I know, I mean, there's only one study or two studies that I know where lithium has been shown to stabilize microtubules, and I'm sure they're going to use the most available lithium around, which would be lithium-7.
Can I just comment on that? Yeah, so there's a kinase GSK3, which in vitro, therapeutic doses of lithium suppress the phosphorylation in a way which is competitive with magnesium, probably along the lines that you were saying, in the microtubules. And we did do the experiment again, switching out lithium-7 for lithium-6, and there was no difference in the suppression of the phosphorylation in that particular case, at least.
Thank you. Last question over here.
In the midst of all this neuronal elitism, I'd like to put a plea in for the 99%, the glia, because nobody here has yet recognized that glia, 9% or so of the cells in the brain, have much of the same anatomical issues and constraints that neurons have without synapses. Matt, there's a calcium depolarization wave that can be propagated in oligodendrocytes and may have an effect on what you are talking about. There are microtubules and tubulin creating the radial architecture of oligodendrocytes and glia, and the myelin sheath that makes it possible for fast transmission, saltatory conduction, rhythm, the music of the neuronal architecture is something that we should probably be paying attention to. Stop looking at just the neurons. We got a whole forest here.
I actually agree with that. Thank you for that comment.
I agree as well.
Thank you.
What I would like to say with the neuron cells is that, or sort of the glial cells, is because they're immune cells, they tend to sense their environment, and they're very labile, or at least when they're active. And so if you were expecting one of these microglia to be migrating to look for something and it's already under anesthesia, you may have it may mess up their lability. And so that's something that we could look at. Thank you.
Thank you, Travis. Thank you, Matthew. Thank you all for being here. I thought that was an excellent session.